So the last topic of today from my part is going to be about molecular diagnostics in beta thalassemia. As I was mentioning at the beginning, the way actually I kind of planned these 3 hours of the content of the talks was to take the students from fundamentals towards an example of a disease, genetic disorder, so as they are able to find out about the content of how a disease can arise, what may be involved in terms of initiating a disease, and the basics in terms of molecular biology and genetics. I will also here touch base upon certain technologies as well. And then I will wrap up by giving 3 different choices of how we can handle or how we can aim finding a cure for this particular disease that I'm going to be talking about, which is the beta thalassemia. Okay, so I would like to take time to say something regarding the genetic disease of hemoglobin in general. So we just basically shortly touch base upon the protein synthesis or translation. This could have been the hemoglobin protein itself. Hemoglobin protein is made up of, or just, let me just go over this and then I'll show you the picture of the hemoglobin protein itself. So the genetic diseases in general, hemoglobin is involved with 2 categories, 2 classes. One of them is the hemoglobinopathies, which means the defects on the protein itself. And the other one is the thalassemias. So this can be beta thalassemia, alpha thalassemia, gamma, globin gene involved thalassemias, delta, whatever. So thalassemias in general is a term that is involved with the gene, the diseases, the mutations that take place on the gene, which gives rise to certain forms of the thalassemias. The word thalassa in Greek means the sea. So at the time when a scientist called Cooley was studying these patients in the Mediterranean basin, that was the main reason why he actually coined this as thalassemia, so anemia of the people living around the Mediterranean basin, shortly, thalassemia. So and then afterwards, he kind of, and other scientists started to come across these kind of genetic disorders because people started to travel. So they didn't localize at original places, such as the Mediterranean basin countries or some of the North African or some Middle Eastern parts. So we tend to get these kind of hemoglobinopathies or thalassemia disorders in Americas, in the world where China, Australia, because people start traveling and there are marriages amongst people with whom the genes are being distributed all over the world. But the important thing is that, to sum up, we need to, when kind of trying to find a cure, or when trying to establish a regimen for prenatal diagnosis, it's always beneficial and advantageous if we know the roots of the people in question. So if we're going to take 2 parents that are in search of a cure or in search of an answer regarding the mutations involved in their genes to do with beta thalassemia, it's always nice to find out where these people come from, whether they're from, for example, Italy, Greece, Turkey, Cyprus, Egypt, China, and other countries that, you know, we started to see these kind of genetic disorders frequently. So once we know these, because of the large number of research in this area, so far there have been actually, one of my slides says more than 200, but nowadays it's like more than 300 different mutations have been reported in the beta globin gene and its vicinity that actually causes these beta thalassemia mutations and the disease itself. So again, I will share with you some pictures where it will show you how the prenatal diagnosis is done, and I will continue to mention at that point. So going back to the hemoglobin protein itself, this represents an adult hemoglobin. In other words, hemoglobin A for adult, HbA. This is made up of 2 chains of alpha and 2 chains of beta genes. So each chain is a representation of the gene itself. So when we talk about alpha chain, we're actually referring to the product of the alpha globin gene. And so the word chain refers to the protein, by the way. So when we talk about the beta chain, we are referring to the product of beta globin gene. So what you see here is that it's you remember when I was talking about the proteins in general, the structures, I said primary, secondary, tertiary, and quaternary. So hemoglobin molecule is a good example for quaternary structure of protein in general. That's what we give as an example, because it is made up of 4 different sections that get together to form the whole molecule of hemoglobin. And it has been calculated that in the mature red blood cells, which are the end point of the red cells, the precursors would be your reticulocytes, where they actually have nuclei in which the transcription takes place, as opposed to the red cells, which are donut-shaped, mature red cells, erythrocytes, which they don't possess nucleus because of the fact that they possess, the nature created them such that in order for them to hold as many proteins, as many hemoglobin proteins as possible, so that the nucleus has disappeared, there is no transcription taking place. It is the end product. It is the final set stage of the cell itself. It has a viability of 120 days. Every 120 days, new ones are being made, okay? And these cells actually are special in terms of being able to carry oxygen, carbon dioxide, nitrogen, and other ions in them, okay? So the main thing that is going to be the subject matter today is being able to carry oxygen in a healthy, in an unhealthy situation. You see from this drawing that in each component of these chains, 2 beta and 2 alpha, there is a region called a heme group where there is an iron molecule onto which the oxygens are being bound. So each hemoglobin molecule is capable of carrying 4 molecules of oxygen. It has been calculated that in red cells, in a single red cell, there's supposed to be 648 million hemoglobin molecules. Can you imagine that? You cannot see these things by naked eye, but under light microscope maybe. So each one of these donut-shaped, healthy, mature red cells are capable of carrying about 648 million hemoglobin molecules. Time that by 4, that tells you how many oxygen molecules a single red cell can carry. In the case of sick pupils, such as beta thalassemia pupils, especially the severe ones, they won't be able to make proper and healthy beta globin chains. So that means that they are going to lack that portion of the hemoglobin, so there won't be binding of oxygen to the beta chains, that is. So that's why they will be kind of lacking this version of the healthy hemoglobin. For that reason, it is required that these people who are actually suffering from beta thalassemia, they would be on transfusion regimens. So I know of some patients from Cyprus that they require to get blood transfusions every 15 days. So this is a very cumbersome process. It is economically cumbersome because it's not enough just to get a donation of blood and then get on with your life. In addition to that, they need to be subcutaneously injected what we call a chelator agent. In that case, desferrioxamine, which actually chelates the excess iron coming out of the from the process where there isn't any healthy beta chains to form hemoglobin, and then the excess iron coming from the alpha chains, they kind of precipitate, and then they form reactive oxygen species, which in turn they can attack your vital organs such as heart, kidney, liver, and whatnot. For that reason, these patients are constantly under treatment in terms of having been most of the time subcutaneously injected this desferrioxamine chelating agent so that they can bind the iron. These are called iron chelators, and then the iron can be excreted to the urine. Otherwise, if they let to have this iron accumulate in their bodies, that is what I just said, reactive oxygen species can arise. So this is not a good thing for the patient. In addition to that, they also have problems with their developmental processes in terms of bones and every other thing that are side effects of the drugs that they have to be injected. Nowadays, there are some companies that actually they produce chelators through like tablet form. Some patients like this, some others don't like it. It's taken orally, and it functions in the same way as the L1, I think it's called, is one of the forms of this orally taken iron chelator. It usually helps to attend to the meetings where the patients also are part of it, so that you can actually hear it from the patient side rather than being involved just in the laboratory. Because if you don't see the patients and if you don't talk to the patients, you don't know what's going on with them other than you defining their mutations or, you know, helping them have transfusions and whatnot. So it's always nice to go hand in hand with the hematologists and molecular biologists so that we can actually find the response in terms of the big question. So what you see in this slide is my cover slide. This just shows you on chromosome number 11, the family of beta globin genes. Obviously, they don't do their jobs on their own. They have to match their correspondence coming from the alpha globin gene families. But today we are not talking about alpha, so we will concentrate on beta. What you see here is series of genes that actually get expressed in developmental, in ontogenetic format, meaning that during the development, you don't jump immediately to the adult form of these genes or the products of the proteins, but you have to go in sequence of how we are created and which genes are first to express themselves. So we start with, you can see on the top right-hand side, chromosome number 11, you see that there is a locus control region right next to that. It's actually the distance is far away, but right, the first gene involved is the embryonic gene epsilon, okay? Each gene here is depicted by a box, but in fact, all of these beta globin family members contain 2 introns and 3 exons. So the numbers of introns and exons are the same, only the sequences are different, but there's a sequence similarity between the A gamma and G gamma genes, except for the A's and G's in terms of amino acids. So in a sequence, epsilon kind of works with theta. Theta is another gene coming from, you can see the right bottom sausage-like thing, which is chromosome number 16, where the location of the alpha globin gene family resides. So theta and epsilon joins, so you get 2 theta, 2 epsilon, which gives rise to hemoglobin epsilon, embryonic hemoglobin. Then with ontogeny, on the left-hand side, you see the graphics. So all these curves in the graphics, obviously, to be able to draw a line or a curve, you need data points, and you join those points, data points, to form the lines. So this is how they are being kind of formed. You see the representation of the loci, in other words, the tissues or the organs where these genes are being expressed, and then they take into action. So it starts with the yolk sac, okay, after the fertilization. And then the embryonic for that matter. Then fetus, the fetal globin gene expression initially goes through the liver, spleen, and then bone marrow takes the major thing into the adults. So adult hemoglobin, as I said, is hemoglobin A, HbA. Embryonic hemoglobin is hemoglobin epsilon, and then the fetal hemoglobin is involved with hemoglobin F, F for fetus. So 2 alpha and 2 gamma globin chains, as you see G gamma, A gamma over there on the left-hand side, the red colored graphics. So these actually, when one increases, the other one slowly starts to increase. When one decreases, the other gene family member kind of tries to take its action and it substitutes the absence of the other one. But unfortunately, in the absence of beta globin chain products, when there is no substitute, such as the epsilon or gamma, then you start seeing these anomalies in terms of thalassaemic situation or anemic situations. So us, the scientists who are involved with this kind of research, are in search of other family members, such as when I was at the NIH in the United States, one of my major work was on the knockout mice models of the epsilon globin gene. I was trying to find out what actually caused the epsilon globin gene to silence. So the genes are already there, they don't disappear. What makes them be silent, when I say silent means that they don't express themselves, what makes them become silent is that there are some transcription factors that actually inhibit their production, which gives hand to the production of the other genes, such as the beta. When beta is being expressed, epsilon is totally turned off. Maybe there is a little 1% or less of the gamma, but genetically there may be some conditions where together with the beta, alpha is also going to be expressed, but this comes in handy when there's a defect in beta and beta is not expressed. So alpha can be, the gamma can be a substitute for beta. So this is what we're looking for, either the epsilon or the gamma. So we're in search of ways, we are in search of drugs that we can actually use these to initiate the expression of the sleeping beauties, such as the epsilon and gamma, because beta is absent, okay? So in two of my students, actually one of my PhD students, we were doing an in silico analysis of the expression or inhibition of the gamma globin gene, and we came up with some theoretical in silico work that we can enhance the expression of gamma by inhibiting some of the actors that actually are transcription factors, for that matter, that actually are silencing these genes when their time is over, because ontology of human development for these genes goes as epsilon, gamma, A gamma, G gamma, and beta. So when beta is normally being expressed, gammas and epsilons are silenced because there is no need for them to continue expressing themselves because there's already a functional gene. But the question comes in when the beta cannot express itself, then we need to find a way to wake up the gammas or to wake up the epsilon. Either way would be handful because it would help us create a hemoglobin molecule under natural conditions in order to initiate the binding of the oxygen so that these people are no longer suffering for the lack of oxygen or the anemic situations. So moving on, that was just the entrance, that like initial big picture presentation. So how is it that these illnesses or diseases come up? So we know that this is considered a rare disease, although we see a lot of, like in Cyprus, it used to be 14% frequency, but because of the law that the Turkish Cypriots have abided by since 1983, all the couples that want to get married have to have their bloods tested for the carrierness of these diseases. So they need to be knowledgeable regarding whether they are carriers, totally normal, or they are sick, obviously. So unless they know this, they are not allowed to get married. They have to have this test, okay? So since then, one of the situations where we can be proud of for the Turkish Cypriots from the North Cyprus is that the sick-born baby frequency is brought down to 0%. This is not true in anywhere of the world, not even in our neighbors in the south. The Greeks, they don't have that. So ours is 0%. When do we get to see beta thalassemia or sickle cell anemic children being born? If people are migrating to Cyprus, who got married outside Cyprus, who are not bound by our rules, you know, they come to the island as migrants and they are already pregnant. They didn't have any test. And all the burden is on the government, because here the government supports these people to help them get the drugs necessary for the chelation therapies. They supply them with blood samples every 15 days or whatnot. So only these kind of people are creating problem for the government nowadays. Otherwise, and they are thinking of introducing new laws so that the government would not be responsible for these people carelessness, that they come and bear these thalassemic or sickle cell anemic kids in this country. Okay, so both the parents needs to be carriers to give rise to a sick kid in terms of 1 out of 4 or 25%. So this drawing here tells you that. So in terms of having a heterozygote born children is not a problem. The only problem may be an anemic situation, and that can be corrected by, you know, getting additional folic acid and iron, whatnot. So otherwise, if the kid is not sick, there is no need to go through any prenatal diagnosis. But again, for us to know whether somebody is going to carries this or not, we need to do the testing. And I did many of these testings while I was an MS and PhD student at Boğaziçi University. So I'll give you a little bit extended information regarding the beta thalassemia. If you look at this table, it kind of summarizes as to, it says that there is a poor network quality, so I hope you guys can hear me. So I'm going to go through these paragraphs quickly and then continue with the explanation. In the early fetal life, the globin chains combine to form healthy hemoglobin. So you get 2 alpha, 2 beta under regular, normal, healthy situations. So this is okay. But in the late postnatal life and adult life, normal hemoglobins can be predominant in terms of the healthy individuals, 2 alpha, 2 beta. So in homozygous beta thalassemias, you can get the decrease or the absence of beta globin gene being expressed. So absence is also effective. Not only can be defective, okay, in terms of if there's a mutation, it has to be subsidized or it has to be substituted by some way, either at a genetic level or with a drug, so as to individual who suffers from this can actually pursue their lives in a healthy format. So what causes the anemic situation or the disease is the excess, as I was saying, alpha globin genes that accumulate and precipitate in the erythroid cells because they don't have a component to bind with to form the healthy hemoglobin molecule. So this excessiveness and then precipitation of the irons in your cells is going to create a situation where reactive oxygen species can arise with the accumulation of iron molecules. So something needs to be done in terms of removing this excess iron. There comes the chelation therapies, there comes the desferrioxamine or other drugs that can be used to get these out of your system. how is it that doctors nowadays can define whether somebody is a carrier or sick? Usually, gel electrophoresis in some of the labs that are readily available, they do gel electrophoresis, but before that, donating a tube of blood can give the doctors an idea in terms of whether somebody is anemic or not, whether their folic acid levels are low or not, whether there is a iron problem coming from the accumulation of iron and whatnot. And there are some indices where actually we can tell from what we call mean corpuscular volume or MCH or other involvement that can be read from a regular blood donation. These can be decisive of whether somebody is carrier or sick. And there are other components in terms of molecular level where we can actually directly go to the DNA level and then test for whatever the mutations are. And thanks to our studies, I was part of the group at Boğaziçi University when I first started doing my master's, that they initiated this reference labs. They collected a lot of blood samples all over Turkey from east to west to north to south. And then we, with my efforts as well, I have screened more than 2,000 patients, individuals, to actually be able to tell what kind of mutations exist in the Turkish population. Well, everybody living in Turkey is Turkish, but everybody's backgrounds may be different. It can be Kurdish, it can be Cherkess, it can be from Iran or from other parts of the world, because Turkey, Anatolia, can be considered as the cradle of civilizations. Whichever civilization came by and conquered the land, they left their genes there. So there's a cocktail of genes where these cocktails can carry different versions of these mutations. And thanks to that, for us geneticists, this is actually an interesting phenomenon. What we can do, we actually study this at the DNA level, and we have found many previously unreported mutations as well as novel mutations. At the time when I was doing my master's and PhD, whenever we found a mutation, we used to celebrate it because it was a big thing. We used to pour our sequencing gels to determine these differences in the DNA sequence by hand. We used to prepare, when I say we used to, I don't like to sound like a dinosaur, but it was like a few decades ago that we used to do this. And it wasn't an easy task to do the laboratory experimentation using radioactivity and whatnot, preparing your own buffers and then pouring those thin gels. And at the end of the day, you might lose your gel by putting it into the oven and it might wrinkle and then everything is down the drain. So if you have done this before, you know what I'm talking about. Nowadays, the younger generation, they don't even bother to use radioactivity. They don't need to actually, because there is fluorometric ways that these four nucleotides of the DNA ACGT can be defined with different colors, and by using colorimetric detection methodologies, using fluorescent sequencing, you can actually do it in a few hours and you can find out whether somebody has a mutation or not. In addition to this, there are kits that have been defined for certain mutations and for certain populations. At the time, I also was a beta, my lab was a beta site, and I was testing some of these kits where they actually spot the mutations on the strips, and then using individual DNAs per strip, we could actually say which mutation belongs to what patient that we are testing. So there are many different ways of doing these things. I wish we had the time, but it might be a case for another conference, maybe in the future, maybe 5th WANA International Conference. Hopefully we will do it here in Cyprus, North Cyprus. I might be able to present some of the data that I obtained at that point with my colleagues from Iran, all over the Iran, like Tehran and whatnot. So what you see in this map here, what you see here is the distribution of different mutations on the map of Turkey Each dot represents a mutation that was identified by my lab when I was doing my PhD thesis and my MS thesis. So these already have been published. The top part of the map just shows you the representation of beta globin gene and on which part, whether it be exons or introns or in promoter region or in poly A tail region, what kind of mutations you expect it to happen, and then you can actually plan accordingly your experiments. So here is another mutation list, frequency mutation list, according to the parts of Turkey where may come in handy when you're doing prenatal diagnosis and you know which groups of mutations you have to deal with. Otherwise, you're going to be looking for a needle in the haystack, okay? It won't be easy for you to identify more than 300 mutations that has been reported so far. So if you narrow down your list, it would be quicker, easier, less expensive, less time consuming, and to the point. Okay, this is why it's important to define the mutation frequencies according to the populations. So this map, again, thanks to the researchers all over the world, just shows us the distribution of not only beta but also alpha thalassemias, the presence of those two different types of diseases. Now I have been talking about prenatal diagnosis, so I'll give you some insight regarding how and where it can be done. So number one is the chorionic villus sampling. In short, we call it CVS. This protocol can be fulfilled between the pregnancy of 9th and 12th weeks of the duration. Okay, so a sample from placenta belonging to baby is taken by an expert medical doctor, and that is delivered to the lab. And it was the case when I was doing it. So I used to get this CVS, and I used to extract DNA from CVS and also samples coming from the mother, father, and most of the cases there was a sick kid in the family, and do the protocol procedure of mutation identification for all the members and unborn baby of the family. And we could determine, sometimes we could get a result where it would give us a sick baby, a heterozygote baby, or totally normal baby. So it would be the decision together with the genetic counselor of the parents whether they would want to, because it's a medical condition, they may decide to go through abortion, okay? Or usually because of the psychological and economical burdens going on from an already present child in the family, parents usually don't prefer to continue the pregnancy. So, you know, they tend to go through abortion. So it is their decision, actually. So the second means of being able to obtain a sample from the pregnant woman would be amniocentesis, drawing some liquid fluid from the amniotic sac. This would be done during the pregnancy weeks of 15th and 20th week. And from here, again, we precipitate these cells and extract DNA, which we know that it is excreted by the baby, and we can actually determine whether the baby is going to be a carrier, normal, or a sick kid. This is a publication by me talking about, in general, the methodologies that can be used using radioactive as well as non-radioactive methods, depending on how your laboratory is set. We were actually set for all of the above, so we did everything under the sun at the time. So you can use, again, this is another topic of a conference, so I'm not going to go into detail, but just to give you an idea, there are means that you can use radioactively, non-radioactively to determine the mutations. If those of you who are interested, I can send them the PDF of this publication so that they can study it from first hand. Here's another map coming from our collaborators from different parts of the world. They actually narrow down the mutations according to the populations, the countries. So it's, in a way, it's under control towards the known mutations. Obviously, you cannot do anything if something is unknown. You have to still do the sequencing and do your search to figure out what the mutation would be that causes the thalassemic situation. One thing that I need to mention at this point is that now that we're talking about mutations, when I was saying the medical doctors, hematologists should go hand in hand with the geneticist or molecular biologist, is that usually the hematologists, they do not do the laboratory part of this process. So us, the molecular biologists, know it better in terms of defining mutations at the DNA level, okay? So why am I saying this? Because there are certain mutations which the babies, unborn babies, may be homozygous for, such as promoter mutations, such as, I'll give you an example, -101 homozygous case, which is not a strong mutation. So it would be like somebody which would be totally normal and someone which is a homozygous for -101 mutation, which would behave the same way. They shouldn't be needing any blood transfusion. But if the doctor is not careful enough and they didn't kind of take advice of the molecular biologist, and they immediately, because in the blood you can see that it might have some anemic situation, blah, blah, blah. So they would figure out that, okay, this is a sick kid or they need transfusion. Once they start the first transfusion, they have to continue, unfortunately. Okay, so this is the case coming from our experience talking with our colleague MDs. So they need to know what the mutations are before they initiate the transfusion regimen. These are some of the other publications, book chapters that I actually have in the public domain. So those of you, again, who are interested, I can share the PDFs of these. Last but not least, I want to touch base on the potential beta thalassemia therapeutics. I put these under 3 different categories. Number 1, the modalities which increase or which reactivates the epsilon gene. Number 2, modalities which reactivates the gamma globin gene. And lastly, gene therapy. This is a new thing. Well, it's been a decade since it's been out, but there are some drawbacks. I will mention what those drawbacks are. Zynteglo is the name of the game in terms of gene therapy. It has been successful, but there is some drawbacks, as I was saying. So this was part of my postdoctoral studies. So what I did was I was trying to create a knockout mice in a region in the genome where the homologous recombination was the most difficult to achieve. I have screened more than 1,000 embryonic stem cells, but we didn't come across any homologously recombined part. The top part shows you the ontogenesis of human. The bottom part shows the ontogenesis of mouse. So I went through a list of different mammals, seeing which one has the highest homology to the human epsilon globin gene, so that I can use that as a model animal, so that I can start experimenting on that. So when you're choosing a model animal for your experimentation, you not only need to kind of take into account how much it's going to cost, you also take into account that how fast the animal is going to have offsprings, how easy it is to handle that animal, such as working with a monkey would be more difficult to work with a mouse. Same thing with cows, same thing with, well, rabbits are okay. But I did a search of like monkeys, rabbits, cows, mice, homology identification. And then I came to the decision that it would be best for me to use mice as my model animal, because the region that I was interested in, like epsilon globin gene of the human, was highly homologous, like 75% homology with the upstream elements of the epsilon globin gene, where I could compare them and then eventually find out about the homologously recombined clones, inject the embryonic stem cells into blastocysts and try to create these mice models to see whether, you know, it would be helpful for me to create these animals and then work on them. But to open a bracket, they have been able to, in the United States, they have been able to create sickle cell mouse. They have been able to treat these sickle cell mouse, but when it came back to the human to use the same analogy for treatment, it didn't work. So the context, we now know that the context is very important. Although these model animals help us understand some of the stuff, it doesn't one-to-one all the time work to cure human diseases. So this is the, endpoint decision that we can say. So now the modalities for gamma globin gene upregulation. So I showed you the human beta globin gene family, and at one point, kind of nearer to the epsilon globin gene, there was a region called locus control region, LCR. This region has hypersensitive sites. I know these things are kind of farther away from what you already know, but I am telling these to you from my own experiments. So I did not read these in a book or from somebody else's. So that's why I'm not using any text. I'm just telling these things to you from my head because I've done these experiments. So hypersensitive regions are part of the regions along the LCR going from 1' to 5', and these are strong enhancers. You know, during the transcription, enhancers are really important to initiate, as the name says, to initiate or enhance the transcription. And enhancers can be upstream or downstream. They can be farther away from the gene of interest. That's why you see this thing as a looping mechanism. So we talk about hemoglobin switching in terms of looping all these LCR regions to go and find a specific promoter region of the globin gene family, either to stop their expression or to upregulate their expression. Okay, so with one of my PhD students who's already, actually he was from Iran too, now he's back to Iran. So he was a mathematician, he wasn't a molecular biologist. I taught him molecular biology, and eventually we met in the middle. So we used in silico analysis in order to be able to find a cure for upregulation of gamma globin gene. And in silico, we did this. We didn't have the chance to do functional analysis, but it can be done in silico by silencing genes, which actually upregulate the beta globin gene regulation. So it would enhance the upregulation of the gamma globin gene. So this was that. And last but not least, the Zynteglo gene therapy. So this was a lentivirus-related clone from the patient's, his or her own stem cells. And then they created these corrected version of the gene of the mutated globin, beta globin, and then they give it back to the patient. What they have found is that out of 41 patients who received this drug called Zynteglo, about 89% was transfusion independent. This is really, really important outcome. They succeeded in terms of stopping the people from getting blood donation. But every test, every drug comes with its drawbacks. And the drawbacks of this drug was that it had a potential risk of leukemia. Obviously, in one hand, you're trying to correct something. On the other hand, you're kind of messing up something else. So it was FDA approved, but now I think it's drawn from the market and they're still trying to correct it somehow to make it less of a side effect thing so that they can, you know, administer it to these patients, because it is very important that, look, they didn't need for 12 months, they did not need any transfusion. I told you, every 15 days, these thalassemics, especially the severe ones, they need blood transfusions. The ones who has received this Zynteglo, they did not need the blood transfusion for 1 year. But again, there is an outcome in terms of potential risks. So unless those risks are eliminated, then it is not correct to use these kind of drugs. And I would like to thank you. It ends my today's session.